for today is to actually finish things we had started almost 3 weeks back now, if not more. Okay, so hopefully all of you will have that hands-on from those from the previous one. Okay, so this is the same hands-on that we're talking about from a while ago. I will increase the font. And where we were last time was we had covered mapping to Callisto. We understand what happens, how is mapping done. We had understood when you actually do the mapping, what is Callisto trying to do, right? And we had started analyzing the data of Callisto. The quick thing that we had shown is overall our correlation is 0.97 with what we found on Geo. Right? So Geo is where you'll find a lot of, for your projects also, you'll find a lot of data from, and this y-axis was coming from counts that are being quantified on Geo. X-axis is Callisto counts. The correlation is 0.97, but we don't stand there. Where we had stopped was this analysis because we didn't have that file and we had to create it, and you can do this when you have time. But what we're going to quickly do is today is we will download that file, which is nothing but the counts of all the data that is collected on Geo. That is what I've done. So you can download this, unzip it, and keep it somewhere, right? So again, all the path that you're seeing here, you should now be in control over where is your data stored and what you should be pointing at, right? So at this point, I think you have done enough assignments to figure out how to do things. So I will not talk about small things, right? If you're getting a read error, just figure it out. It's not that hard. This is a path which exists on my machine. It might not exist on your machine. So you figure out how and where your path is, right? So for small things, we will not stop now, because I think you've done enough assignments to not run into those issues anymore, okay? So here I'm just reading the counts, and again, I get an error, but that's just because I'm starting from there. All my libraries are being imported in this section, which is here. So just below analysis of Callisto counts, I would just run that first. And this is what we were getting from Geo. This was for one sample. This is for this particular sample. We had downloaded this data, if you recall, and we had read it. What this file that you can download from, again, the course website, all I did was just went to this one, and you can download a zip, unzip it, keep it somewhere. And wherever you keep it, come back to it and just provided that path, right? So I have a directory called data, and inside I have my bulk and all my counts are here. This is the file. So wherever it is, you can do things like, I think there's a way to copy path. Yeah. So you can copy this particular path here, come back and paste here, right? So it only copies the path to the folder. You will have to still do it, right? So again, you figure out where your file exists. My file exists here. Hopefully that's not very difficult to figure out. It's just a file path. This is what that data frame looks like. It has gene name, and the column names are different samples. Everyone with me, people who have laptops? Okay, people who don't have laptops, what are we going to do? You can sit and watch. That also works, but because this is online, so you can go back. But I think today I had posted that you should bring your laptops. Okay, everyone with me so far? Okay. So we had also downloaded the metadata file, and if you don't have it, it's fine. Well, actually, it's not fine. But at some point, you would have downloaded the metadata file. Does everyone have the metadata file? Does everyone? So also, my sincere request, right? You want to laugh, you should laugh your heart out. I will never stop you. Please don't disturb the class. That's my only request. You're all mature guys here. Mature folks here. I don't even feel like lecturing you on this. This is the last time I'm doing this. Next time I see you, you are out of this class. And I'm very serious about this. It's very simple. You just are out of this class. Okay. So we're going to read the metadata. Everyone has the metadata file. If not, I can also upload it. I'm going to do it. If you recall, you will need this, right? So we had done this for, it's an exercise that we had done using PySRADB. I had asked you to run this on your terminal, so you must have run it at some point. If not, you can run it. So for example, I have PySRADB installed. You must have done it in your environment. So we call that environment DH607. I might not have it, but this is actually I have it. So I will run this file. If it is not there, you can always install. Make it this bigger as well. So my environment, the reason I cannot show you everything in my environment is because on my Mac it doesn't work. So I will log on to other server. But all you have to do is very simple. Actually, I will just deactivate this. But you should keep your environment. And what you have to do is just type this command, which will give you this metadata file. I will strongly encourage you to have access to PySRADB because you will need it for your project as well. Okay. This command is here. I'm not doing anything which is not there. Like I'm just copy pasting this command, and that way you will have this metadata file. What does this metadata file have? So the metadata file has different columns. The first column is what's called your SRR number. The second one is your study accession and other things that are required. Everyone has a metadata file now. If not, like if you have a Aman, don't have it? You have it? Okay. Anyone who's having trouble getting the metadata file? I can upload it, but since you're going to do projects, right? Time is to level up, and I'm not doing anything pristine. I'm not doing anything novel. Literally copy pasted file, a line from there. I had shown you how I figured out what I should do, right? So this SRP is the project that I got the data from. That was a GeoID, and each GeoID will also have an SRP ID, right? So to calculate the metadata, you need that SRP number. So our GSE, again, just so that you know how, this is the GSE number. You can always go to this GSE, you can Google it. Make sure sometimes the Google results are not correct. Make sure the number is correct. We could go there, and at the bottom you will find this SRP number, right? So that's what the SRP number was. And what I'm essentially asking you to do is just get the metadata, save it to a TSP file. Okay. Okay, so this is what we had done actually in the previous class. That's why I assume all of you will have it, right? So if you have puzzled faces now, that's not my mistake. we did it in the class. Okay. So now we are going to actually do the actual stuff. So we have the counts.csv file. The counts looks like this. Each number column here is a GSM ID, right? Now the reason I'm asking you to actually get the metadata is because it is just a number. We don't know what this sample is associated with. Is it a control? Is it a healthy sample? Is it male, female, whatever, right? Like whatever can go wrong here. So that information comes from the metadata file. That's why we require that metadata file. And that's why we are going to process here. So I should run this again. In the second, this code block, what I'm doing is to do the rest of the analysis, your row names of your counts matrix should be a gene or a gene name. That's what I'm doing here. I'm making it unique because sometimes gene names can be repeated, right? So before you do this, your row names cannot be, they have to be unique. And there is a very simple function in R to do this. So the way this function works is, if I give it, it will make the first number A, and for the remaining gene, it will just append a .1, right? Because you want the row names to be unique. If you don't do this, R will complain that you're trying to set something which is not unique. So it is a very quick function to make a vector unique. Okay. Okay. So I'm going to read the metadata. The metadata you've already seen. This is what it looks like. It has multiple columns. This is where you have your stuff, right? So this is the title. It has liver patient 97. There is some description here, and what is the strategy, what is the accession, and so on. So you'll have to do some data munching to figure out that this belongs to this GSM, because that's the name of your column, right? So that's why it will work in this case, but any other dataset that you download will have its own format of how things are stored. So that's something that you will have to figure out, right? The goal should be clear. The goal is that I want the column names. I want to know the column names which are in my counts matrix. What is the metadata associated with it? Metadata is what stage or whether they are control, cancer, or whatever, right? That will be in a different data frame. So that's this data frame. So there is a column called stage in this metadata, which encodes my stage information. So we have 10 control samples, 138 early, and 68 moderate samples. We will have to set the row names of metadata to match the call names of counts. Okay. So the metadata needs to have, it has a column called experiment alias, right? This is what that experiment alias is. This is the same name, or this is very similar-ish to, or actually this is the same as what your column name of the counts are, right? Why that is done? So I need to set the row names of the metadata to the experiment alias. That's what I'm doing here. Make sure you don't bring in information from any other data frame here. And what I'm doing, I'm assigning the row names of this data frame to be an experiment alias, because I will use it later on. It will become clear in just a bit. And finally, I'm assigning this. I'm creating a new column in the metadata frame, which is called sample name. You can call it whatever, which is just experiment alias, right? You'll see in just a bit why I do that. Okay, so our object or our counts values are ready for doing downstream analysis, right? So we have, what do we have? We have, let's call it sample 1. So some 296 samples. This is our counts matrix, and then we have a metadata, which is nothing but the metadata is associated with the samples. Okay, so these are the row names of this matrix. Then what do you store in metadata? It's a very free flowing thing, right? So it could be stage of the disease, It could be a date when the sample was taken. It could be age. Any other factor that you think, right? Sex, gender, any other thing that you can think about that will vary across these different samples. Usually it will have the sex sample and usually not the gender one. But these are things that you would ideally have in most cases. What is our goal? Our goal was, how does a different stage of disease impact the gene expression, right? So if the disease stage is controlled, early and moderate, I want to know what is the difference between, let's say, control and early, or I want to know what is the difference between control and moderate, or between early and moderate. The reason I want the other columns is because I want to adjust for the fact that across these different categories, the age and the sex could be different as well. So we want to account for that overall in the design. Right? That's why we have it. To do this, we are going to make use of this tool called DH607. DH607 is a tool which we will study very briefly. But very simply, this tool tries to, given accounts matrix and a metadata, it allows you to identify what genes are differentially expressed using a negative binomial statistical model. It will become clear once I start my lecture. This is just all the information that you need at this step. It allows you to do an exploratory analysis on your data. It allows you to perform normalization, and it allows you to perform differential expression, which is our goal for this exercise. Okay, everyone with me so far? So all of this stuff here is not magical. It would have definitely come from somewhere, right? So DH607 has a very good manual. If you just search for DH607, its manual is extremely long, but it's a worthwhile exercise to actually go over this in detail, especially those of you who are going to work with bulk data in cases versus controls. Very broadly, it tries to show you, given different scenarios, how should differentially expressed genes or biomarkers be calculated, what should be an exploratory analysis. We will do some of it in very brief, but of course we cannot do the entire manual. That's something which you should go back and try to do. Okay. So I'm going to create the what's called the DH607 data set. This variable is called DDS. That's what this represents. It takes three values. One is counts. What is the corresponding metadata for that counts? And what is the design matrix, right? This design stores, essentially tells this model, statistical model, that things here that I'm most interested in is that things are going to vary because of stage. So if you have ever studied a linear model, any linear model is in the statistical terminology written as y tilde x, which means y is a function of x. Similarly, here I'm saying that counts and the metadata, it's a function of the stage. That's my value, that's my covariate column that I'm interested in. Okay. This can always be revised, so not something to worry about, but you need to specify a certain design. And the reason I'm specifying stage is because that is what my interest is in, and also that is the only usable column, by the way, in this metadata, right? Let's look at what other columns does this metadata have. There's nothing very specific otherwise. There is experiment description. All the organism name is going to be the same. In other words, there is no other factor here which will help me decide, or which will help me justify that including that particular covariate will lead to changes in my samples, right? All of these are other just links and so on. There's nothing extremely meaningful. There is some source name, so that is early versus moderate and so on. That's already captured in your stage. There is something called NAS score, and there is something called fibrosis stage. Ideally, we could include this in our design, right? This is a clinical factor which says that fibrosis stage is something that will come from your from an ultrasound scan, that these patients were also went under an ultrasound scan. I could in principle use it, And if when you actually start to do projects, you should do it. You should include as many possible variables that can explain the differences. The reason I'm not doing this is because I'm going to just show you what happens if you start from a very simple model and how you can always make it more complicated as you go along. Okay. Then there is something called group in paper, so that also captures your overall stage. These are all NAFLD samples. There is some control here. That control is already captured in the stage. So there is no explicit requirement of capturing the disease as a column because it's already captured in the stage, and that's what I did in my exercise. But beyond stage, fibrosis stage and NASH score, there is nothing in particular that in this case you would want to include in your design. What you include in your design is sources of variation which will lead to changes across the columns in terms of the counts. Okay. Any questions at this point? Yeah, so the NASH score is a measure of how severe is your fatty liver. It is a function of how much deposition has happened of the fat. We haven't covered the biology in this class for that, and we will not cover it, but that's basically a proxy of how severe your steatosis is for liver. Okay, that's a clinical parameter that they have assigned. So a good question then would be that, once you do your biomarker analysis, which of these biomarkers actually inform your clinical parameters, right? So some of these clinical parameters would come from a different scan. Then is it something that you also see molecularly? Because that's coming from a, let's say, ultrasound, then that in ultrasound all you're doing is imaging. Does imaging actually inform, or does imaging actually reciprocate in terms of what you see at the molecular level? That's something which you could, in principle, ask with these kind of data sets. All of that is going to be based on the idea that whatever clinical staging has been done is close to real, like that's correct, right? If that is of course wrong, then you will not see anything. And that's why the exploratory analysis which we are going to do now is going to be useful. Okay? So here, there is some filtering required. So before we do this, let's play around with the data a bit, right? So our counts matrix has 62,000 genes and 216 columns. The easiest things that you could do is you could count what is the mean expression of this gene, right? So what I did is I just calculated what is the row mean of my counts, and I can just plot. There is a lot of genes which have very low expression, and of course there are a lot of genes which have very high expression. Right? So on a log 10 scale, you'll see most of these values are very, very small. I just calculated the row means, and I'm plotting the overall expression distribution of the mean expression, and this is what it is. Last class I told you that there are two things that you should always measure given a counts matrix. What is the other thing? This is the mean of the rows. This is showing you that a lot of genes are mostly very lowly expressed. Right? There are few genes which are highly expressed, but most of genes have very low expression overall. What other things would you want to know about this matrix? Okay. You did violent plot. Okay. Violent plot, what should we do with? Okay, that's good. So we should, for example, one of the things that you would want to be, that you would be interested in is just look at how is the total number of molecules one second. Oh, sorry. How is the total number of molecules different across the 2216 samples, right? So I'm plotting a histogram of the total molecules, call sums. I'm not doing call means, you can do call means also. It will give you a similar trend plot. It is showing you that approximately the minimum number of molecules you have in one sample is 10 to the power 7.2, and the extreme is 10 to the power 7.6. In other words, it's a very similar-ish sample, right? Like the total sequencing depth of these samples is between 1.5 to 3 or 15 to 30 million unique fragments within each sample. right? So these will sum up to 216 samples, and this shows you 15 to 30 million reads, 15 to 30 million fragments overall. Any other thing that you would want to know? How does it help actually, right? So this is just showing you that before you do any sort of, what the fun thing that you'll have to account for is that even to begin with, there is technical differences. The total number of molecules across all the 216 samples is not same, right? So there's nothing explicitly that you're going to do with this information. This is for your use case or your learning that these things are not constant. Like there is differences there. Let me quickly also show you what the distribution of so this package you might not have. If you don't have matrix stats, you can always install any package using install.package. You can always do this. I'm not going to do it because I already have it. But what I want to plot is this. I want to plot how does the okay, so you actually this is a good way to teach. So here I'm running into an error, right? But this error is actually telling me what is wrong. Argument x. This is where I'm getting into an error. Argument x must be a matrix or a vector. And the reason I'm getting that error is because counts is not a matrix; it's a data frame. So this will happen to you also. It doesn't mean that for everything you have to go to Google. It actually tells you here, or whatever your LLM, it actually is telling you the error, right? It's telling you why it has an error. You should be able to resolve this by convert. So how would you fix it? Counts as a data frame. What the input to this function that should be is a matrix. So in R, you can understand or you can figure out what is the data type of a particular variable by running class on it. So if I run class on this count, it tells me type of data frame. What should be the input to row variance? It should be a matrix or a vector. In this case, I cannot make it a vector because it's actually a 62,000 cross 216 vector matrix, a data frame. And what I want is I want to calculate the row variances. I want to calculate what is the variance displayed by each gene. So what I could do is I could cast this as a matrix. So that is not a very unintuitive function. It's called as.matrix, which means that cast my data frame as a matrix. And if you run this, right, so this is what it looks like. It says you can't really see a lot going on because there is an extreme outlier all the way at the top, which has a very high mean and a very high variance. Now, the next step that we're going to do in DH607 is we're going to stabilize the variance, and in the class we will discuss what is stabilization. But the problem, if you want to work with this data and you want to find out what are differentially expressed genes, given the fact that the ones which are highly expressed also has a very high variance, if you run any statistical test, these are the genes that are going to stand out, just because of the pure fact that these genes are simply high expressed. It doesn't mean whether they will have they are different between control or early. Just because these are very highly expressed, any statistical model will flag them to be differentially expressed. And that is what we want to prevent, right? We want every gene to be weighed in a way where we have removed the effect of the variance relationship. So this in general is not good, right? If the variance has a relationship to mean, and That's why base plotting is not good. You can do, I'm going to use this function called qplot. Qplot comes from ggplot2. This still doesn't look as decipherable because these are things that you would ideally plot on the log axis. So that is very easy to do using scale log x, and you see what is going on, right? So now on both the log scale, you can see that as the mean increases, the variance increases, right? Again, in your case of your Gaussian, the mean and the variance are not related. But if I run any statistical test on, if you do your t-test using this data, you will start to get only highly expressed genes as your variable ones, as the ones which are differentially expressed, because these have very high variance overall. Right? So the model, any model that you need to run, you should make sure that the data satisfies these assumptions. This does not satisfy the assumption of your Gaussian, because the mean is a function of your variance. We can make it look like Gaussian, and that's what we'll do in the class when we actually just begin our class in just a short bit. But anything that is not clear right now. Is the problem clear? So in Gaussian, whenever you do t-test, z-test, whatever, right, there's an inherent assumption that there is a mu and there is a sigma square. This is your mu, and this is a measure of sigma square. And if my genes follow a Gaussian distribution, that sigma square can be anything. They're not constrained by what is my mu. This mu could have been higher, and I could still have got that sigma square. So if you plot if your genes are following Gaussian distribution, and if you plot this curve, what curve will you get? Assuming all the genes have the same variance. If all the genes follow a Gaussian distribution with the same variance, this is called homoscedastic assumption. Homoscedastic means the variances are equal across all the observations. What plot would you get? Straight line. Yeah, you get a straight line, which is your sigma square constant. Whatever that constant is, you don't know. In this case, what you're seeing is you're seeing a curve which looks like this. Right. So this A means whatever the spelling is. You can figure out whatever the right spelling is. But it's a curve which follows a relationship between variance and mean. And whenever you see heteroscedasticity, particularly when the variance is a function of mean, that's usually not good. Usually you would want to remove that effect before you analyze your data. Okay, can happen in multiple contexts. Not just gene expression. You take your most volatile stocks, for example. Higher stocks will have higher volatility as well. Like higher price stocks might have higher volatility. You can see a very similar relationship there. Okay. Anyway, this part is clear. Okay. So then we will have to do some filtering. So this is required because when this engine is going to process our data and fit statistical model, it is not a big fan of seeing a lot of zeros. It likes to see things which are somewhat complete. So this step tells that only retain genes which have at least 10 as its row sum. Okay? As long as in the 216 samples, if the sum of each gene is 10, at least 10, then only retain those genes, right? So I'm going to run this. What was the original size of our data? Some 60,000, right? And now it's down to 19,000. So there were a lot of zeros, which also, that's why I drew the previous plot, where we just did the histogram of the row means. there are a lot of samples which have a lot of negative values, right? So that is something which it should be this. Sorry. So we don't want. So whenever you're doing log, it's always a good idea to add something to it so that that minimum thing is 0. So you have a reference to compare this against. So this, we have very lowly expressed genes. We want to remove them, and we only have now 19,000, which are going to be which we're going to work with. Okay, we'll proceed. What is the next step that we're going to do? In the next step that we're going to do is before we do any differential expression analysis, we will try to just do a very preliminary test on how does and what does our data look like. So if you just, at this step, if you want to just do differential expression, you can, and let's do it.
So I'm confused, like you showed the graph mean and variance. Which step is handling that?
Yeah, good question. We'll show you in just a bit. So when I run this magic command called DH607, it actually prints multiple things, right? So it's estimating size factors. We have covered size factors when we covered about the DH607 normalization, right? Which what it tries to do is it assumes that 50% of the genes nothing is going to happen. You take the geometric mean of each single gene, that becomes your reference. You come back and you weigh how much higher is each gene's expression with that pseudo reference, and then for each sample you figure out a size factor. So let's do this step by step, right? So this is the command, by the way, to answer your question. This is the command which is doing everything at once. It is estimating size factors. So let's look at what are our size factors, right? So this is again a command in DH607. You can print size factors, which is DDS. For each sample, it's calculating a value which needs to be divided by, each sample needs to be divided by, which will try to bring in all the counts that you have at the same scale. Remember, the problem is that we don't sum to the same scale, right? Like the problem is that our initial distribution that we have is going to be very, very different in terms of the call sum. So let's try to see if we can plot this. So I'll just do a hist. So a lot of them have a size factor of 1, which means that you're keeping it as it is. Some of them have a size factor which is less. Some of them have a size factor which is higher. Okay. So what does it do after this step, right? In this step, it stores the normalized values which have been normalized and variance stabilized in So if you want to understand, one second, what each of these values are. So when I ran DH607, it created an object like this, right? So on the right panel, you will see that there's an object which you can actually try to understand what does it look like. It's like a data frame, but it stores multiple different things. It stores what is my design. It stores what is my dispersion. It stores what are my row range, and so on. And then somewhere it will store my actual data, right? So it's a very complicated object, but it's something which you can always print, right? So whatever is at the rate, in most cases you can access it by what's called an accessor function. So if I do at the rate, it will tell me what all are possible. So if I do assays, it tells me that it has assays of type of called data, and I can keep doing that. This is where my raw counts are stored. So I did at the rate data and then $count. So at the rate is the accessor for accessing a particular slot, and in this inside this slot it stores accounts, right? So some of this is very technical, which you don't have to worry about, because you can actually get away without actually knowing this. But what I want to show you is where are these values stored, right? So the normalized data is stored in this object. If you just do DDS, assay DDS, it stores the counts object, right? So let's do, for example, I can always see for T-span 6 and for GSM, what is my count value for this? Okay, this is 1725. This is what I get from my assay as well. So assay DDS is where the counts are stored. It stores the normalized data as well. I'll show you in just a bit. But you can always access your data which is stored in all part of your DDS object, right? So in this case, they are exactly the same. You can always try with any other values that you want. And you see that all the counts are stored in this particular assay data. So when you run this function, inside this function, what it is doing is it's doing multiple things. It's trying to calculate the dispersion factor. It's trying to estimate these dispersions, and it is going to store as part of that object, right? So all of this is available as one different slot in this object. Now, when we do remaining of the functions, when we do exploratory analysis, and when we call some other functions, all these calculations which need to be only done once are reused to do the remaining calculations. So let's do the first calculation here. So the first function that we are going to do is we are going to calculate what's called a variance stabilized version, right? So remember, we have a variance mean relationship, which we showed that variance is a function of mean. Here I am calculating it to calculate a transform where the variance is no longer a function of mean. It's called a variance stabilization transformation, VST. I am storing this in a variable called VSD, and we're going to very quickly see what does it look like now after that your variance stabilized. Overall, what is this function going to, what mathematically it is doing, we'll cover in the lecture in just a bit. But this is what this looks like, right? So this is your original raw counts mean variance relationship. You can do the same thing with the VST, and notice what is the distribution of your variance now after that you've stabilized it with respect to mean. What does it look like? More or less straight line, not still there yet, but this is the best that we can get access to right now. There are other ways to sort of think about why it's not doing a good job, but this is what it looks like, right? So from there, from the previous one which we had in raw counts data, we have now been able to brought it up to here. The reason this is important is because if you want to do any exploratory analysis with any dimensionality reduction method, none of the dimensionality reduction methods, for example, there is one interesting one which is called PCA, principal component analysis. Its underlying assumption is of homoscedasticity, which means that the variance is not a function of mean. That's why there is a standardization step required there. We have not covered PCA in this class yet. We will do it in the lecture. But a lot of dimensionality reduction techniques assume that there is no function between your variance and your mean of your data. So if I had fight it the counts data as it was previously to PCA, I'll get very weird results. But now that I have stabilized this, it's of the same, the dimension of this is same as your original counts matrix. But the values that you have, right? So I had shown you this. I will now show you this. Those values have been transformed so that these are not, these are now variance stabilized. Okay? From the same sample, same gene. If you have access to this, you will want to run what's called PCA, right? We will study formally what is PCA, but for now I'm going to just use the function and run this, and this is what it looks like. Okay. Okay, tell me, what do you think is going on? Each dot is one sample. I'm coloring it based on the group that was there in the paper. I can also color it based on the stage. So this is with group in paper. I can also just do it here. What do you think is going on? Is it a good plot? Is it a bad plot? What do you make out of it? 12% of the variance is captured along the first dimension. 11% is captured along the second dimension. What do you notice here, and what would you have liked to see? Each dot is one sample.
There is probably something else at play, not visible.
Okay, why?
Because the early is also getting split into two clusters.
Hmm.
Moderate also getting split.
Okay, so there is some sort of confounding variable here. This is why you should do exploratory analysis, right? Like, I can trust them on face value. You can run the fanciest machine learning algorithm here. It will give you something, but does it actually mean anything? That will come from spending some time doing this exploratory analysis. So, in an ideal world, you will want to see that there is information in all the three things, right? So, control, early, and moderate hopefully should have made three different clusters. In a lot of these settings, it will not because sometimes the changes are very subtle, like they're not as extreme where you will start to form totally three different groups. So they will be very subtle, but forget about that. Something else is going on for sure, because there is this line in PCA which separates these two groups out. So PCA is a fantastic technique. In fact, one of the questions usually I ask people in different settings is what would be the input source so that you will get this kind of data distribution? What do you think is happening in the input data source structure that you get a pattern like this? We have not covered PCA in the class, but intuitively what PCA tries to do is it tries to look for a possible rotation in your data such that the variance of the projected points along that projection is maximized. How many of you have not seen PCA or do not know what is PCA? Okay, fair enough. That's fine. I will cover it, just that some of them have an overlap with the other class, so that's why it's been covered there. Okay, here, right? So think of it as a dimensionality reduction tool, which is going to reduce your dimensionality. What is your original dimensionality? Let's start the question there.
Is there a difference of interest?
Okay.
Just seeing it seems like it's just an addition of an intercept into it.
Okay, good.
What is that in the data?
Okay, so good, very good. So there is definitely an intercept difference, right? Like PC is not regression, but one way to think about PC is regression. You can definitely see, in other words, there are two sources of information that is seeping into your data, right? So there has to be something there. So original dimension, right? So we started from some 19K into 216, but here I am visualizing my 216 samples. So 216 samples and its original dimensionality is 19,000. There is no way and no technique in this world which can help you visualize it. So what we do is, even before I do the analysis, I can bring it down to a 2D picture or a 3D picture. And the way or the technique that we are using to do that is PCA. We will cover it very quickly in the class, what intuitively what it tries to do. Okay, that's what you're seeing here. And what it tells us is the reason is, if there is differences, you will want to see it here itself. So there is definitely something going on, right? Now we will try to, what will you do next? Your goal was different. Your goal was to figure out what is different between control, early, and moderate. But we cannot do that yet because there is a confounding factor which is explaining something weird going on in my data. Now if I do control versus early, my actual signal is going to get masked out. The reason being that if I just do control, or if I do early versus moderate, right, which will be blue versus green, a lot of these differences might just be coming because these two samples are two different clusters which might not have equal number of points. So if in the cluster 2 you have more green, and in the cluster 1 if you have more blue, and if you don't adjust for it explicitly, it will start to show differences in your differential expression. So before you proceed, you want to figure out what explains this. So now is your turn to come up with hypothesis. What explains this? What could likely explain this?
DH6 or something else.
Okay. Age, sex, collection time, maybe who processed the sample. Something definitely technical, right? Which should have been captured but is not there, right? So we can try, right? Like we can try all different things. You don't have to believe me. You can always go and what was that column? Let's do fibrosis stage. I think it'll complain, but let's see. Don't see any pattern. What is the other one? NAS score. Don't see any other pattern at all, right? So I don't have any, and none of the stuff, or of course I can do disease, but that's going to look weird as well, right? There are a lot more disease samples as compared to control samples, but again, that doesn't explain it. There has to be definitely something. So age, how will you test if this is coming from age? If age is the covariate. But you need some, it's not there in the table. This can happen with your project. This will happen with your project. A lot of your projects, you'll run into this issue. I'm not saying that you have to do projects only in RNA-seq, but any other technology, any other method, this will happen. Where do you go from here? Age, how will you guess? Okay, let's go to the paper. So, where is it? Paper, right? Well. Okay, let's go to the paper. Within the discovery cohort, 38.4 were female. The average age was 55.2 plus minus 11 years. Average body mass index was 33.5 plus minus 6.7, and 60.7% had type 2 diabetes and whatever. Supplementary table 1. Table 1 and supplementary.